Welcome to the video on process hold time study in pharmaceutical industry. It is also called as hold time study. Hold time study is performed onto the pivotal batches or exhibit batches whenever the product is being developed for the specific markets. Nowadays, many of the regulatory authorities require hold time study and its data before product approval. Many of the times the whole time study batches are required to be taken separately, and some of the times the exhibit batches material is kept in simulated bulk packs as in process material for whole time study. There are different ways to perform the whole time study depending on to the formulation design and the criticality of the formulation. Specific product will have different hold time. If the product is sensitive to environmental conditions or microbial contamination, that time the hold time will be less. Let's start with the video for process hold time study in pharma industry. It is also known as hold time study. In this video we are going to learn what is the hold time study, why it is important, and we'll see the topic in depth with exploration of process hold times, specifically in the pharmaceutical product manufacturing. It is abbreviated as PHT, that is process hold time, or simply it is called as hold time. And it is the time period during which the intermediate materials, raw materials, and bulk materials are held or hold under specific in process packs for specific time under specific storage conditions for the next processing. For example, if you are working onto a tablet manufacturing process, then you may be working with direct compression, roller compaction, or wet granulation process. Simply, if you are working onto the wet granulation process, then the materials which are required to be held, it may be for testing purpose or it may be for the next processing purpose. For examples, as in the tablet manufacturing, first the stage is manufacturing of the lubricated blend, then is the compression, and then is the coating process if the tablet is coated. Generally, the intermediate stage materials will have specifications. For example, lubricated blend is required to be tested for assay and blend uniformity. Also, it is tested for bulk density, tap density, particle size distribution, LOD, moisture content, assay, and blend uniformity. These are the testing. So, the bulk lubricated material is required to be stored before compression. Then, after compression, the core tablets are required to be tested for assay, dissolution, and other testing before going into the coating process. Then the coated tablets are required to be tested before these go into the packaging process. So at these stages the process is hold, and the time for this hold period is called as process hold time, or simply it is a hold time. Hold time studies are essential for ensuring the product quality, safety, and efficacy. If you hold the material for longer time, and if it absorbs moisture or it undergoes degradation, then it will have impact onto the product safety and efficacy. So the transitioning between manufacturing steps is the hold time. We'll see in this video, does the time between one step and the next count is a hold time. This is the question. Intermediates with no registered specifications. If the intermediate material is not having any specification, for example, dried granules or milled granules, these are not having the in-process specifications. How do you manage and monitor material that doesn't have defined stability specification during their processing? This raises concern about ensuring consistency in all steps. So it is not like that only the lubricated blend will have specification or dried granules will not have specification. Binder solution will not have specification. Coating solution, coating dispersion will not have in process specification. But some intermediate materials are with the specifications like lubricated blend, core tablets, coated tablets. And these specifications are called as in-process specifications. Sometimes machine breakdown may be there, because of which we have to wait for the machine correction or the process correction. So some time, because of the breakdowns, because of some issues in the routine manufacturing, the material is hold. That's why the hold time studies are also important. Then sometimes because of some production emergency or priority changes, there may be chances of holding one batch in some in-process stage and taking another batch in the continuous manufacturing. So that's why the hold time studies are essential. Some challenges are there in validation for hold time. Cumulative versus separate intervals or cumulative versus separate hold times. For example, if you are making a tablet with wet granulation, you are holding a lubricated blend for, let's say, 30 days or 15 days. You are also holding core tablet for 15 days. You are also holding coated tablet for 15 days. So total hold time will be 45 days. Like PHT is equal to PHT1 plus PHT2 plus PHTN. So n number of hold times will be there. So should the validation consider total hold time across multiple steps, or it is a single or separate hold time interval? So should each step in the manufacturing process be validated separately, assessing each hold time individually? Validation must ensure that the overall product stability is maintained regardless of how the time is distributed. So if you start a batch and you complete the batch in 1 month, so that total 1 month will be the whole time, whether you are distributing it into the lubricated blend or core tablet or coated tablet. Many of the times, separate whole time batches are required to be taken, because as per my experience and understanding about whole time in the pharmaceutical industry, I think and I believe that as per my understanding and experience, if you are holding lubricated blend for 15 days and then it is going into the compression process, sometimes compression process itself may become difficult, because the lubricated blend flow will get affected, its moisture content may get affected, it may show segregation, there are chances of segregation. So separate batches are required to be taken for establishing the hold time. Hold the lubricated blend for 15 days, go for compression, do the compression, understand the effect of hold time on compression process, understand the effect on physical properties, chemical properties of core tablets, then hold the core tablets and go for coating. Hold the core tablets for some days or weeks as per the protocol, then go for packaging. And compare these batches data with the batches manufactured without hold time. This will give the clear-cut idea about validation challenges and separate versus cumulative hold time. So when to start the validation? It is a question like what is 24-hour rule? Many organizations start validation after 24 hours. Is it sufficient? And why 24 hours is the starting point? So it is not like that only the 24 hours is the start time. We can design the protocol and we can do the whole time studies. Many of the organizations get queries from regulatory authorities for these whole time studies. Why whole time study is critical and why PhD validation is crucial. So the main challenge is chemical stability and microbial risk. Prolonged hold times may lead to chemical degradation, which affect assay and impurity profile, which impact safety and efficacy. Sometimes because of the moisture, light, oxidation, hydrolysis, the potency can be reduced or impurity can be generated. Sometimes because of the microbial contamination, there may be microbial load, increase in the microbial load, bacterial or fungal growth. So mainly these microbial risks are mainly related to vaccines and injectable formulations, sterile formulations. Storage challenges for single-use systems. So single-use systems may introduce extractables and leachables which could negatively impact the product quality. Buffer solution and stability. Extended hold times or extended hold required validation of the chemical and microbial stability of any buffer or solution used during the manufacturing. So whenever it comes to the parenteral formulations, injectable formulations, sterile formulations, the whole time validation study become very critical and crucial. Understanding importance of process whole time validation. PhD validation is essential for ensuring that all materials maintain their quality and regulatory compliance even if held for extended periods. Failure to properly validate PhD can result in compromised product quality, delayed launches, and non-compliance with the industry regulations. So, maintaining the quality of the formulation, compliance with the regulatory requirements, and the timelines, launch of the product or filing of the product, query response of the product is affected because of the process hold time. What is the path forward or what is missing? So regulations versus practical guidelines. There are very limited guidelines for the whole time validation, and whole time validation varies from product to product and organization to organization. Regulations generally highlight what needs to be validated, but there is limited clarity on how to validate PhD effective in various scenarios. Whether you are considering the separate PhD or cumulative PhD. When to begin the validation of specific type of formulations like biological products, pharmaceutical products, sterile products, chemical products, or nutraceutical products as well. Now, let's have a brief idea about this PhD. So, PhD is crucial for industry, experts, and regulatory bodies to provide more detailed practical guidance on managing and validating process hold times to ensure both the consistency and safety. Many organizations remove the samples from the ongoing batches of exhibit or pivotal batches and they keep the material and they test the material for quality. But when you hold the entire batch and then you perform the next process step, that becomes the real world scenario or real time activity. Hold the lubricated blend in the tablet formulation. Do compression after hold time. Then hold the core tablets. Do coating if required, if it is there in the formulation. Then hold the coated tablet before packaging. This will give the real scenario or real time study. It will indicate whether the hold time affects the compression process or not, whether the core tablet hold time affects the coating or not, whether the coated tablet hold time affects the finished product quality or not. Many organizations are required to provide the justifications. So hold time study nowadays become very critical. Process hold times are essential to maintain the product stability, quality and safety during the manufacturing. And process hold time or hold time study and its validation is critical for both the chemical and microbiological safety. So I hope this video will give you clear understanding about the hold time study in the pharmaceutical manufacturing. It is also called as process hold time study. And if you come across any specific guideline, please comment and I will try to go through those guidelines, as presently there are less number of guidelines on this topic are available. Thank you for watching the video and let's meet you soon with such kind of informative video. Thank you.